Today we're talking about Chapter 6, Section 2, which is the process of meiosis. And you can see our key concept here. During meiosis, we start with diploid cells. Remember, diploid means two sets of chromosomes. They go through 2 rounds of cell division and results in 4 haploid cells, which you can see here at the end. Again, haploid meaning 1 set of chromosomes in each cell. So let's talk about how that happens. The 2 stages of meiosis, we call them meiosis 1 and meiosis 2. Meiosis 1 is always going to start with a diploid cell. In your body, this is only going to happen in cells that we call germ cells. Germ cells are cells that undergo meiosis to produce gametes. And during meiosis 1, as you can see in this animation here, the 2 chromosomes from a homologous pair are going to become separated into different daughter cells. So you can see it going through the stages here. Don't worry if you don't get it all right now. We have a diagram a little bit later with more of the details in it. But here you can see chromosomes condense, nuclear membrane goes away. We see those homologous pairs line up side by side, then they get separated, and then we can see the formation of new nuclei. Again, this is part of meiosis 1. Now, during meiosis 1, we have a process called crossing over. And this is when 2 homologous chromosomes come together to form what we call a tetrad. Tetra means 4, and each homologous chromosome has 2 sister chromatids, so all 4 chromatids together. That's where the name tetrad comes from. When those tetrads are together, you see segments from one chromatid switching places with another chromatid. And this is what we call crossing over. This is an important source of genetic variation. It is not a mutation. It's not a mistake. It happens frequently, and it happens for a specific purpose. That purpose is to increase genetic variation. So if you look at this diagram here, you see a light green chromosome and a dark green chromosome. They are going to become entangled with each other and actually switch places. So whatever alleles were on the dark green chromosome are now a part of this light green sister chromatid, making this sister chromatid different from its other light green sister here. And then the same thing on the other chromosome. The light green alleles have now become a part of this chromatid. So now we see that all 4 of those chromatids are going to have a slightly different DNA sequence. Now we mentioned 2 rounds of cell division. So let's think about what happens in that second round. Meiosis 1, remember, was about separating homologous chromosomes. But now in meiosis 2, this is when we have the separation of sister chromatids. The end result of meiosis 2 is 4 genetically unique daughter cells. And by genetically unique, I mean they have a different DNA sequence. All 4 of those daughter cells are haploid. So let's look at the animation here. 2 cells from meiosis 1, we have the lining up of those chromosomes in a single file line, separation of chromatids, and then the formation of new nuclei and cytokinesis. So meiosis 1 happened right after DNA had been replicated. Remember, that occurs in the S phase of interphase. Meiosis 1 was responsible for dividing homologous chromosomes in 4 phases: prophase, metaphase, anaphase, telophase. Now we put a 1 after each phase name so that we know it's happening in meiosis 1. So here's your chance to really look at the details of what's happening in each one of those phases. We're starting with a diploid cell. How do I know it's diploid? Well, we have 2 big chromosomes and 2 small chromosomes, right? We have a purple and a red chromosome from the mother. We have a green and a blue chromosome from the father. In prophase 1, we see the same things that we were seeing during mitosis: the breakdown of the nuclear membrane. We're going to see the chromosomes are forming that really condensed structure. We're going to see spindle fibers starting to assemble. But the one thing that's different here in prophase 1 of meiosis compared to mitosis is that this is when we see crossing over. Crossing over occurs during prophase 1 of meiosis. That's a key point. Crossing over can only occur during prophase 1. Now we're going to move on to metaphase 1. In metaphase, remember, chromosomes are lining up in the middle of the cell along the equator of the cell. Well, notice how they are arranged. They're still arranged as homologous pairs. So it's homologous pairs of chromosomes lining up in the center of the cell if we're talking about metaphase 1 of meiosis. This is now going to take us into anaphase 1. Since we had a homologous pair line up side by side, look at what's getting separated in anaphase. The entire X-shaped duplicated chromosome is being pulled to one side, and then its homologous partner, this purple X-shaped duplicated chromosome, is being pulled to the opposite side. Same for the red and the blue pair. The entire red duplicated chromosome is separated from that entire blue duplicated chromosome. So in telophase 1, we're forming that new nuclear membrane. The DNA is going to relax back into chromatin. We've got the breakdown of the spindle fiber. But notice that we only have one large and one small chromosome in each of these daughter cells. That means that each of these daughter cells only have one set of chromosomes, meaning they are haploid at this stage. Right? Two sets of chromosomes here. This was a diploid cell. They have now, at the end of meiosis 1, become haploid. Now let's talk about the steps of meiosis 2. Big picture for meiosis 2. We are dividing sister chromatids. Again, 4 phases: prophase, metaphase, anaphase, telophase. Now we're going to put a 2 after each name. DNA is not replicated between meiosis 1 and meiosis 2. That only happened at the very beginning during interphase. It does not happen for a second time. So here's our diagrams of meiosis 2. We're going to start with prophase 2. And you will notice that this entire process here is pretty much mitosis. So look at prophase 2. We have DNA condensing into chromosomes. We have spindle fibers forming. We have nuclear membrane disappearing. All of that happened in mitosis. Now take a look at metaphase 2. The spindle fibers are moving those chromosomes into position. And what position are they moving them into? It's a single file line along the equator of the cell, just like we saw in mitosis. In anaphase 2, now we see the separation of sister chromatids. Those spindle fibers are pulling each X-shaped chromosome apart, and the sister chromatids are moving to opposite ends of the cell. And finally, in telophase 2, we have the formation of a nuclear membrane around each set of chromosomes. We have the breakdown of the spindle fiber, and the chromosomes are going to relax back into chromatin. So again, start to finish, meiosis 2 looks exactly like what we saw in mitosis. So let's recap the ways that meiosis is different from mitosis, because there's some pretty significant differences. One major difference is meiosis has 2 rounds of cell division. Mitosis only had a single division. In mitosis, we never had the pairing up of homologous chromosomes. We never had crossing over take place. That is only a process that happens during meiosis. At the end of meiosis, we have haploid cells, or cells with only one set of chromosomes. Mitosis, however, resulted in diploid cells. We started and ended with diploid cells, and remember, in mitosis, those cells were genetically identical. So here's a diagram that's comparing the two. We've talked about what most of these things are. The only thing I haven't really mentioned thus far is the role in reproduction. So mitosis is involved in asexual reproduction because we're just making more cells that are just like the ones that are already there. Whereas meiosis is involved in sexual reproduction. Remember, sexual reproduction involves combining DNA from two different parents. So in order to combine those cells together, you have to have a way to make cells that only have half the normal amount of DNA. And that's what we see in meiosis. So obviously, this is only going to happen when a cell needs to reproduce, to make new offspring for sexual reproduction, whereas mitosis happens all throughout an organism's lifetime whenever it needs to grow or make new cells because of an injury, anything like that. Now, those haploid cells that are a process of meiosis, they have to finish developing in order to form mature gametes. So let's talk a little bit about how that happens. First of all, this is called gametogenesis. Gameto for gamete, genesis means to make something or to begin something. So gametogenesis is the production of gametes. It's going to be slightly different between females and males. In males, obviously we're making sperm. They have to be able to swim. So in order to mature from the end of meiosis to a actual mature sperm cell, they become more streamlined and they become motile, meaning they can swim. They develop flagella. And the contribution of sperm to the embryo is really just the DNA. So it doesn't contribute any cytoplasm, it doesn't contribute any other organelles, it's just contributing its DNA. So let's look at what that looks like. In the production of sperm, we have a germ cell that's diploid. It undergoes meiosis to make haploid cells. We get 4 haploid cells at the end of meiosis, and they mature into sperm cells with flagella, and they have a streamlined shape so that they are able to swim. Now, eggs are a little bit different. Egg cells are going to contribute DNA, of course, but also the cytoplasm and the organelles that an embryo needs. So that means that the division of cytoplasm has to be a little uneven. An egg cell wants to have as much cytoplasm, which contains organelles, as possible. So let's look at that in the form of a picture. We have the germ cell here undergoing meiosis. After meiosis 1, notice that uneven division of cytoplasm. The cytoplasm and organelles are all concentrated into this one cell here. The other cell is what we call a polar body. You can see that at the bottom. A polar body doesn't have enough cytoplasm to survive, so this actually becomes reabsorbed by the body, and this is the only cell that will undergo meiosis 2. In meiosis 2, we again have the uneven division of cytoplasm to make one really big egg cell that contains almost all of the cytoplasm and organelles, and one small polar body that gets reabsorbed. And this is because this egg cell is essentially going to become an entire other organism. It needs to have cytoplasm, it needs to have organelles to support that organism's development. So the final thing that I'm going to leave you with here is the section quiz for Chapter 6, Section 2. After what we have discussed here in this video, you should be able to read through these questions and come up with the answers. If you can't, or if you have any questions about them, please make a note of that and make sure you ask about that in class.